In this video, we shall look into the history of DNA and its dimensions. DNA was first isolated by the Swiss scientist Friedrich Meiser in 1869. He extracted it from the nucleus of white blood cells and named it nuclein, which was later known as DNA. Rosalind Franklin and Maurice Wilkins took X-ray crystallographic photographs of DNA. In 1953, James Watson and Francis Crick, using these images, they discovered the three-dimensional double helical structure of DNA. It looked like this. A double-stranded DNA molecule has a backbone of deoxyribose sugar and phosphate group. Nitrogenous bases bond to one another, forming a base pair, which appears like a rung of the DNA ladder. Let's look into the dimensions of a DNA molecule. The diameter of the double helix is 2 nanometers. The double helical strand has minor grooves and major grooves. One major and minor groove makes one helical turn. The length of one helical turn is 3.4 nanometers and there are 10 base pairs in one turn. So what will be the distance between two adjacent base pairs? We can find the distance by dividing 3.4 by 10, which is 0.34 nanometers. So the distance between two adjacent base pairs is 0.34 nanometers. It's time for a question. If all the DNA from the nucleus was pulled out and stretched, it would reach approximately 2 meters in length. So how do you think it fits in such a tiny nucleus? Coiling the DNA helps reduce its volume, and this helps it fit in the nucleus. There are some specialized proteins that help in wrapping the DNA molecule. These are known as histone proteins. A total of 5 histone proteins are involved in this process. They are H1, H2A, H2B, H3 and H4. DNA is wrapped around these proteins to form structures called nucleosomes. This highly coiled DNA ultimately forms a chromosome. Here are the assignment questions. What is the total number of chromosomes in a human cell? Find out the total number of chromosomes in a pea plant, frog, pigeon, and shark.